Imagine a woman who lived in the first half of the 1900s, an African-American woman. She learned about her cancer in her early 30s, faced the terror of death, and shortly thereafter departed from us. However, this departure was different from other deaths because as she died, she also declared her immortality. Because that woman, Henrietta Lacks, who touches the lives of millions today, changed the face of science and medicine by providing immortal cells to humanity forever. Before delving into the topic, let me remind you, if you enjoy the video, please click the like button and show it on YouTube. Also, if you subscribe, you can support us. First, I want to talk a bit about how Henrietta Lacks' cancer cells were obtained because there are some ethical questions to be addressed in this matter. Lacks, as I mentioned, was diagnosed with cervical cancer in her 30s. But remember, she was an African American, and it's unnecessary to dwell on the difficulties faced by a black woman from a poor family, especially when Jim Crow laws, causing racial segregation, were in effect in the United States. Fortunately, at least one of the important hospitals of that time, Johns Hopkins Hospital, had a section dedicated to black patients, and Henrietta Lacks found an opportunity for treatment there. Frankly, I don't want to compare the service provided to blacks with that given to whites at the hospital. That's another topic for discussion. But let me briefly touch on the experiences of our young cancer mother. Essentially, she went to the hospital a bit late. Even relatives encouraged her to go to the hospital, but in vain. According to some sources, she didn't want to see a doctor because she feared having her uterus removed, while others say she was generally hesitant. However, when the symptoms worsened, she understood the seriousness of the situation and reluctantly underwent a medical examination. After being diagnosed with cancer, she received the standard radium treatment of that time. In other words, radiation was applied to her body. Even the source of radiation, radium, was sometimes placed in special containers and inserted into the cervix, a kind of primitive cancer treatment with radioactivity, we can say. Of course, radioactive metals can be lethal to healthy cells, causing mutations. However, they can also kill cancer cells and slow down their growth. Also, during the ongoing radium treatment, healthy and cancerous tissue samples were taken from her without her consent during operations. That's where the main story begins. Unauthorized samples were taken, and two famous cell biologists of the time, George Gay and Margaret Gay, who were conducting research on cancer and viruses, were sent to work on them. Because one of the main goals of this duo was to grow cell cultures outside the body. Let me explain. Cell culture is the cultivation of cells obtained from plants and animals under controlled conditions. Likewise, a cell line refers to the independent replication of a group of cells outside the organism. Normally, healthy human cells have a very limited lifespan because each of them has an internal control system. However, when it comes to understanding how cells work, the causes of diseases, or developing and testing theories for treatments, preserving and growing human cells in a laboratory environment plays an important role. And when Henrietta Lacks' unauthorized cancer cells fell into the hands of George Otto and Margaret Gray, they immediately realized something: these cells were immortal. They were dividing and multiplying repeatedly. Imagine this: in 1950, no laboratory could grow human cells for more than a few weeks. Now, in your hands, you have cells that grow robustly for 24 hours. You've collected samples for decades without discrimination based on religion, race, or gender. And finally, you are achieving your goal. You can now perform many experiments on these cells that you couldn't do on a human. You simply add the first two letters of the name and surname of the person whose cancerous tissue you took. HeLa, however, unfortunately, all these developments were kept secret from Henrietta Lacks. She had already passed away after some operations. So, what made HeLa special? I would like to touch on the biology part a bit without forcing you, and to help you understand scientifically what happened, let me mention some concepts and tasks. In the cells of living organisms, there is DNA, the genetic material where all genetic information is stored. This is a term you're probably familiar with from middle and high school. We call the shortened and layered structure of DNA, known for its double helix, a chromosome. So, in fact, DNA and chromosomes, which we call structures, are the same, just different in shape. Well, since we're trying to understand HeLa cells now, let's continue with human cells and take a slightly more complex look at the structure of DNA. In humans, there are telomeric structures at the ends of chromosomes that repeat and preserve genetic information. In every DNA replication, in every cell division, these telomeres shorten. Yes, as you know, they shorten and cause the chromosome to shrink. When telomeres don't shorten, aging and the processes of death progress proportionally. Therefore, the telomere regions in our DNA, shortening with each cell division, bring us closer to death. But at the same time, there is an enzyme called telomerase in our bodies, which prevents the shortening of telomeres. While continuously dividing cells and reproductive cells show telomerase activity, somatic cells, the cells that make up our body structures, do not have telomerase. Here, scientists associate HeLa cells' immortality precisely with these telomere structures. It is thought that the presence of an active telomerase, which gradually prevents the shortening of telomeres in cells, allows cell division to continue for a very long time, preventing cellular aging. Believe me, it's tremendous for humanity to have such a cell line. Why? Because one of the biggest problems of the 1950s was still caused by polio viruses, which lead to childhood polio. This disease can seriously affect children's nervous systems, causing symptoms such as vomiting, fever, fatigue, reflex loss, muscle spasms, and even paralysis. And in the 1950s, the vaccines developed against polio, which were normally being tested in cells taken from monkeys, were an extremely expensive procedure, especially if you consider that you kill cells during the process. Ideal cells, sensitive to the infection caused by the polio virus during testing, were necessary. But she shouldn't be killed by it either. The rapidly multiplying HeLa cells fit perfectly into both of these criteria, and thanks to them, we developed vaccines for polio and largely got rid of this plague. So, is that all its benefit? Of course not. In the 60s, HeLa cells were sent into space to determine the long-term effects of space travel on living cells and tissues. We used them to understand how much damage a nuclear explosion could cause to human cells. In 1965, the first human-animal hybrid cell was produced using these cells. In the same year, HeLa assisted in gene mapping studies. In 1986, it was used in studies on HIV, and in 1993 on tuberculosis. Can't we use HeLa for COVID-19 research, or even for understanding the effects of the cosmetics and skin care products people use every day? And, of course, let's not forget that we need it to discover potential cancer treatments. How important is that? But, what about the unauthorized use of HeLa cells that we've benefited from in over 100,000 articles worldwide, and their later commercial use? What should we do about it? Well, after George G. discovered H, he first shared this information with his colleagues. Then, he sent HeLa cells to researchers in Texas, India, New York, the Netherlands, and many other places. He even went to different institutions to demonstrate culture creation techniques. Not only that, but he also helped establish new laboratories, doing all of this mostly for free and without obtaining a patent for H cells. So, we must not overlook his contribution to science and humanity. Perhaps he saved the lives of millions of people. Johns Hopkins Hospital has been saying for years, "We haven't made money from this, we haven't sent a bill." However, these immortal cells were later used for commercial purposes by different institutions and are still being used. Well, is there no moral problem here? Now, first, we need to understand the conditions of that era. First of all, in America at that time, taking tissue samples without consent was common. Scientists of that era considered it permissible for the advancement of science. Besides, there wasn't exactly a legal regulation about obtaining consent from the patient. In this case, it might be a bit strange to retrospectively judge the doctors who performed H's operation and George G. about why they took those tissues without permission. I'm not saying what they did is right, it's definitely not ethical. But why should we judge and accuse them without a clear purpose? They called it HeLa, but initially didn't share the full name. This information was leaked first, and later, they admitted to the incident. Perhaps they refrained from telling her family because of ethical considerations, or maybe fear of the family asking for money, I don't know, or perhaps they withdrew from something, but I don't know what. Imagine you're on the verge of death, leaving behind 5 children, and groundbreaking research is being done with the samples taken from you. They don't tell you or your family about it. After a while, companies making money from this research emerge. Mind you, a regular family might say, what does it matter to us? But when the first discovery was made, blood samples were taken from the family without explaining why. In some research conducted by different individuals and institutions years later, some genetic information about the family was obtained. Now, isn't all of this a violation of rights? It seems it is to some extent, because recently, decisions were made that justified what happened. Although it didn't benefit them financially, at least a small compensation decision could have been made, and society's trust in medical research might have been restored. Wouldn't that be a bad thing? I leave the decision to you for today. That's all from me. If you liked the video, don't forget to like and subscribe. Until we meet again, goodbye.